Okay, everybody. It's 2 minutes past 12 now, and we've got a nice collection of participants. Over 70 people have joined. Sure, some others will join as we're going. So today we're going to have a seminar by Professor Christine Beveridge, who I'll introduce soon. First of all, I will introduce myself. If you could advance the slide, Christine. So I'm Professor Mark Cooper. I'm the chair of crop improvement in the Centre of Crop Science in the Queensland Alliance for Agriculture and Food Innovation. So I'll be your host for today and moderating the presentation and the question and answer session at the end. So it's my pleasure to do the acknowledgement of country. Myself and the University of Queensland acknowledges the traditional owners and their custodianship of the lands on which we all participate and meet from. We all pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country, and we recognise their valuable contributions to Australian and global society. So if we advance there. So in terms of some housekeeping matters, the seminar is scheduled to run from 12 until 1. And at the conclusion of the seminar, which may run from 45 to 50 minutes, we will have a question and answer session. And so within the seminar platform, you'll find that there's a Q&A button on your screen. So if you have any questions, please enter your questions into the Q&A section. And what I'll do at the end is I will actually check for the questions and I'll present the questions for Christine to respond to. Please don't enter your questions into the chat button as we won't be looking there. So now it's my pleasure to introduce Professor Christine Beveridge, who I've had the great pleasure to work with since I've joined the University of Queensland, and a lot of collaborative activities in the Centre of Excellence for Plant Success in Agriculture and Nature. And I'm sure Christine will talk about this. Christine is known to a lot of us for her really leading scientific work on the studies of the genetic and molecular biological physiological controls of shoot branching. And if you ever spend some time with Christine, you'll find that one of her favorite topics is talking about plant hormones and sugars, and particularly her favorite discovery is that of the involvement of strigolactones as a plant hormone and how sugars actually act like plant hormones in a signaling network to promote branching. And apical dominance is, she'll clarify to you that apical dominance is not just about auxins and cytokinins. So I'm sure that we're going to hear a lot of exciting updates on Christine's research in this seminar. Important things to note before Christine starts her seminar is that Christine has recently taken up the role as the Centre Director for the Centre of Horticultural Science in coffee. So congratulations to Christine, and look forward to listening to an inaugurating seminar in her new role. Christine's a fellow of the Australian Academy of Science, and she has a very distinguished position as an ARC Georgina Sweet Laureate Fellow, as well as being a highly cited researcher, and has a strong commitment to leadership, equity, and ensuring that we have a very healthy and diverse scientific community. So with that brief introduction, Christine, I'll hand it over to you and look forward to your presentation.
Yep. Thanks very much, Mark. It was very kind of you for that somewhat lengthy introduction. It has been a pleasure working with you as well over the years since you came back to UQ. So this talk is titled Inspired by Plant Development Research. and it's really about how we can design networks and pipelines that can be used for prediction across a range of activities in agriculture. So hopefully I'll convince you that we are well on track to do those things. And I'll just thank Mark for his acknowledgement of country. I too acknowledge the traditional owners and their custodianship of the lands on which we meet. And I also want to express my commitment to diversity and inclusion. I love this slide from Coffee that talks about this issue. And I really believe that diversity of thought and the inclusion of diverse people, diverse thinkers, different backgrounds and cultures and so on really do enrich our ability to innovate and to do a good job at what we do in science. And I'd like to start actually unusually with acknowledgement to the lab. There's a number of people here I'd really like to thank, and some of their projects I'm actually not going to cover today, on branching and flowering, on how initial bud release is prompted, and how abscisic acid is involved in shoot branching, some work in horticulture, crops going on in the group. But I won't be touching on those particular items in this talk. But I will start off with a section on why I do, what inspires me about plant development research in terms of a way of thinking for how we might consider doing modelling. And in the second part of the talk is around designing and networks and pipelines that, as I said, can be used for prediction in a range of activities in agriculture. So for the first part of the talk, yes, so shoot branching has been the thing I've focused my mind on throughout my career to date. And, this is a picture of a mango plant that Inigo sent me. It's been pruned, and you can see the shoots that grow out as a consequence of that pruning, and that process is a really, really fundamental process that goes on in many horticultural crops, and there's many issues that come with it. And also, of course, in broadacre crops such as sorghum, tillering is a really important component of yield, as it is in wheat. But it's a trait that's really hugely influenced by environment and, of course, by management, pruning, but also nutrient supply, heat of drought. All these things influence the architecture of the plant and consequently influence its yield. And really it's those things that attracted me to come to Coffee to think about how I could make a contribution. But before I get onto that, just want to focus on this plant development work. And yes, as I started my PhD, which was well after this date, it's actually before I was born, just so you know. Basically the theory was that auxin and cytokinin were opposing signals involved in shoot branching. Most of this was through apical dominance studies where they kind of cut off the shoot tip and the idea is auxin depletes, cytokinin goes up, and that's how come you get branching. When I started, there was no genetic analysis at all really of branching, and I was very fortunate to be given a range of mutants, including this one from my supervisor. And I was able to show through simple grafting experiments and hormone measurements that there was a novel graft transmissible signal that's produced in the root and importantly also in the shoot of plants. And that was an unknown signal. And other labs soon after also realised that, in Arabidopsis, for example, there's another novel signal. And similar genes encoded carotenoid cleavage enzymes that was discovered about 2003, and very soon after, actually the biosynthetic pathway of another hormone family was found to be carotenoid derived, and that's what led us to the discovery of strigolactones as the branching hormone in 2008. So subsequent to that, we've been able to use the knowledge of that hormone and all the genetics to start nutting out how all these signals interact. But because pruning is so important for agriculture, well, at least for horticulture, I wanted to focus a little bit in this talk about decapitation, about the pruning response and what's really going on there. And, you know, it was known for some time in the late 90s and 2000s that auxin depletion moves about 1 centimetre per hour. And when you sort of sit there for a moment with a bit of time to think, and you think, hang on, if I've got a plant that's 20 centimetres tall, will it really take 20 hours for bud outgrowth to happen at the bottom if this has got to get down here? And so we did some experiments with Jim Hannon, and folks of you may know him from coffee, to actually test whether or not this auxin depletion actually is able to move quickly enough. In this experiment, the dots are radioactive auxin supplied. The black and white are decapitated intact model of auxin transport. The light grey is modelled auxin depletion, and the dark grey is endogenous auxin levels. These are endogenous too after auxin depletion. But the point is, that's what's happening at the top of the plant. We're up to 4 hours. Buds start growing out the top. And then at the bottom, all of a sudden, the bud actually starts to grow out. So the timing, whichever way you look at it, this is an incredibly detailed modelling exercise to demonstrate there's no way that even the very front depletion of auxin could actually be sufficient to trigger that bud to grow out. And so those experiments and others demonstrate that actually auxin is not the trigger, that is the depletion thereof that is causing these basal branches to grow out. So the obvious other candidate, and of course that had been in the literature in the 60s, was sugar. Of course, sugar's not as cool as a plant hormone, so I think that's probably why it got ignored for so long. But if you actually look at the transport of sugar using carbon-11, it's actually extremely rapid, and we find that sugar redistribution in the plant after you remove the sink, which is the shoot tip produces auxin, but it's also a sink for, you know, much of the photostimulate produced by the shoot. So the sucrose actually moves extremely rapidly, so 1 metre per hour, not 1 centimetre, and that redistribution happens very widely spread across the plant. So instead we have this model where we've got sucrose accumulating extremely rapidly after decapitation, and that led us to propose that actually sucrose is a signal that is inducing bud outgrowth. And I've already put some other symbols in there, of which there are papers in the literature now that we've contributed to that talk about these relationships. But just wanted to highlight some genetic information by Francois Barbier and Franzi Fitchner, who were here at UQ and working on the sugar signalling pathways involved in shoot branching. So coming back to this idea that the sucrose is moving redistributed so quickly after decapitation, you just sort of scratch your head again and think, but okay, well, what about the cytokinins? I mean, are they really auxin regulated in this system, or maybe it's the sugars? Oh, yeah. So, you know, same experiment, but this time we're going to measure the levels of cytokinins in the stem at the base of the plant instead of just measuring the outgrowth of the bud. And actually, within 1 hour, we see a significant increase in cytokine level in the stem tissue and in the bud tissue. And of course, that increases dramatically over time. But remember, it takes, 20 centimetre tall plant. It's going to take about 20 hours for the auxin depletion to reach there. We're getting these changes within like 1 hour. And I guess that's the whole point about thinking about experimental design, isn't it? Because if you design a plant that's only tiny, like, this big, well, you might get the correlation. But if you design a big plant, you don't get the correlation, But sort of 101, you've got to try and disprove your hypothesis. And in so doing, you discover something completely new. And, again, this audience being interested in horticulture well, just broadly plant science. It's interesting to note that also they're happening in the bud are the well-known hormones that are often associated with bud burst in perennial crops, We've got gibberellins here, which are also changing really rapidly in the bud. What this plot is showing is time, so 1 hour through to 24 hour with the different coloured time points. And then we have gibberellin level on this axis and auxin level on this axis. So you're seeing this beautiful correlation, which is well established in the literature around auxin regulating GA levels. And that work, that correlative work's been done by John Ross, backed up with a lot of mutant studies as well. And what they've shown over the years is that it takes a bit of time for that gibberellin to actually kick in and actually lead to phenotypic outcomes, which means that that GA is increasing rapidly because later on it's going to be important for the sustained growth of the bud once it's released. So coming back to our diagram here, we can then modify it. We've got our sugars, they're interacting with the cytokines, but also, and we're just guessing because it happens so fast, the auxin level goes up in the bud, that's enhancing the GA, it's promoting bud release, which is a very first stage of bud outgrowth, and then the GA is really important for the sustained growth. Another work I'm not showing would associate auxin transport with this sustained growth phase. So, yeah, that gives a bit of a feeling of sort of how we go about learning how plants regulate developmental processes such as branching. And, you know, it's great to have these networks, and I'll show you a bigger version later. But what real impact is that having on agriculture? So if my parents say to me, you know, Christine, I mean, you spend a lot of taxpayers' money, you know, what's the use of it? I'm like, well, I get these great papers, Mum and Dad, and they'll be sitting in lectures forever. And they're like, well, where's your real benefit? And I'm like, well, people are using these genes, you know, to associate the gene with maybe a QTL that allows them to know that perhaps a strigolactone gene is involved in, you know, maybe it's tillering, maybe it's response to parasitic weeds, which is another thing that strigolactone is involved in. But when I honestly think about it, my heart is really around the network itself. It's around how these components actually interact with one another. That's where the real power of these networks is. It's not just a list of genes, it's how things interact with one another. And really, that interaction's very difficult to be utilised by people outside of a very specific domain of shoot branching. It's very hard to just pass that knowledge on. So within the Centre of Excellence, our goal is to understand how to take prior knowledge of networks and make them useful for a range of activities in agriculture. And so I'm going to talk to you about some of the work that I've been associated with within the centre in trying to achieve that. It's just a small part of that work, but I hope you'll enjoy the story. And it kind of starts thinking about, you know, the problem that a plant breeder such as Mark, who hopefully is still paying attention, might be thinking about. If he's wanting to muck around with bud outgrowth, he's wanting to modify it in a breeding population, perhaps thinking about how to minimise the variation across environments, or perhaps just create a line that might tiller a lot in a favourable environment, for example, for sorghum. It's very difficult to, you know, when you've got all these different loci that are actually tracking their way to that phenotype through a whole bunch of other things, to actually be able to disentangle that in a breeding pipeline. And so what we're trying to do is to bring the networks in between. And so the talk I'm going to present from now on will have address this from three points of view. The first is, how can we actually extract that knowledge of the networks into a format that Mark could use? For example, a list of mathematical equations. He does maths, he can look at those equations and make use of them, but me just showing him a diagram or telling him stuff's probably not very helpful. So that's the first way, design a way of extracting and validating mathematical descriptions. And then the second way is, okay, that's great, you've got a list of mathematical descriptions, but can you actually make them useful in a breeding pipeline? And then thirdly, you know, can we actually do this at scale in an automated fashion? And so we can go to AI to see if we can achieve that. So for the first one, Nicole Fortuna has been driving research to figure out how to extract and validate mathematical descriptions of networks, and I'll emphasise from diverse sources of knowledge. So, a network that story I just presented comes from a whole bunch of different experiments and different papers, and there's many, many, many other things. So my network has to come from diverse sources of knowledge, and not just my research, but many others. And we think that it should utilise the developmental concept of change relative to wild type or control. So you have a control plant, you decavitate it. You have a wild type plant, you have a mutation, and in both cases we're looking at the phenotype in response to decavitation, or the phenotype in response to that mutation. So it's always change relative to the wild type. And I think that's a very important and key concept that we can leverage in modelling, because if we think of the wild type, we can think of a steady state, and that's very important. So that would mean that we want to capture relationships of increase or decrease in response to a treatment, which means we need to have inhibition and promotion as integral parts of the network. Also, in order for it to be useful, I think it does need to be scalable, so that we can have multiple traits. We can have, as new knowledge comes along, we can add it to our model and not have to create a whole new model from scratch every time we need to build in some new knowledge. Importantly, it has to be very deeply evidence-based, and that comes with transparency. So we need to be able to understand every single part of it and where the evidence comes from, because otherwise, how's Mark going to ever trust me that this network's going to be any good? I think that also enables us to test current understanding, which of course is important for plant development research in itself, and it has to be able to be shared with non-experts so that you can pass it on without needing any prior knowledge. So I want to just introduce you to this simple model, which is a redrawing of the model I've shown you before with sucrose, cytokinin, auxin, and strigolactone. It's a mathematical model that was generated by Jessica Berthowit in a collaboration with us in New Phytologist. And she made a nice differential equation model. And this is just a part of the diagram which shows the fit of the simulation in the lines with the observed data. That model had 19 parameters, so it's quite a highly parameterized model. And so Brody Lawson, he's very interested in how you can use Bayesian methods to identify parameters and observe what alternative possibilities might also fit the network. And he's just got two examples here. One here on the left with the similar Berthowit parameters, another one on the right which has different parameters, and you can see the shape with the inhibition here being quite a different shape between the two parameter sets, but both of them actually fit the data, which are the coloured circles. So that really has implications if we want to predict into new environments or new situations. When we highly parameterise something, it really influences what the model is going to look like outside of the space upon which the observed data is obtained. And of course, that's going to be really problematic too if I haven't told you yet, but this model that we presented actually works equally across diverse species like rice, Arabidopsis and garden pea. And it's highly unlikely that any parameters that we utilise or even data sets would be able to utilise the same parameters. So we think it's a good idea to see if we can move away from that. And so that's something that we've achieved in our branching modelling approach. Now, I also wanted to point out too this crazy set of, not crazy, it's a set of data from that same paper, and all of these experiments, you don't need to know any details, but all of these experiments, just to show you, they're all wild and wonderful experiments in pea and rice, pea and rose, and in vitro, intact plants, adding hormone, adding sugar, There's a bit of mutant work in there. All of that work was helping to produce this diagram, and then that diagram was validated against one data set to produce those parameters. And what we think is that instead we'd like to use this diagram to actually validate against all of this data, so that everything has to fit all of the data, not just the one data set that we use to validate. And if we can achieve that, then we can realise that we are able to take a modelling approach that would be able to make knowledge, utilise knowledge from diverse sources. So different publications, not just different experiments within one, but across different publications. So that was a task, just a small task that Nicole was sort of took up the mantle for in starting a postdoc with a Centre of Excellence. And so she came up with this PSoup R package. I'm not sure if this is a logo forevermore, but I'm sticking with it for now. But anyhow, so the point of PSoup is that it's focused on predicting relative change, as I mentioned before. It can be used to predict against any data sets, as I've explained the need for in the previous slides. And it's modelling at a steady state. So we're modelling the network in the wild type or control situation. And the benefit of that is that nothing's changing if we think about that over time, over a short period of time where you can imagine a plant's in a steady state. And that means that we can build onto the network without introducing any change to any other part of the network at the steady state. So that's really important. It also enables a ready analysis of relative change to that control. And the network, and I'm using the knowledge graph, the old-fashioned term here, it's a diagram that describes the knowledge that we currently know, and Nicole's designed it such that those networks can be automatically translated into a mathematical description. And this approach is, let's say, intermediate between ordinary differential equations and Boolean networks. So to do that, Nicole utilised the SBGNAF notation, which is a standard notational literature which explains simulation, inhibition, necessarily stimulants, and so forth. And so the diagram I've been showing you earlier can be redrawn like this, with an integrator as a theoretical node upon which information is integrated to regulate branching. So we've still got our auxin, strigolactone, cytokine, and sucrose. And this is the, if you like, the mathematical formulation that is used. So we can think for each node, let's say this node here, there will be stimulation, inhibition, there's necessary stimulation and a modifier. And it's the sum of all the things that make it go up, divided by the sum of all the things that make it go down, with a caveat around the necessary stimulation. And the modifier is used, that's a place where the user comes in. And for example, the user can come in and say, hang on, I'm going to make this gene a mutation. I'm going to make it a mutant. I'm going to completely knock that out. Or I'm going to add some hormone to the system or something like that. So we have modifiers associated with any gene, any node that we choose. And that's the way that we can modify our network and then see how things change. So back to our simple model. We use PSoup then to produce a set of algebraic equations. That's an automated process based on the structure of the diagram. And then we can use PSoup to simulate biological outcomes relative to, model outcomes relative to biological data. So we represent the biological data in terms of increase or decrease relative to baselines, which are shown in white. And then we compare the model to those biological changes. Just removing all the controls and also only identifying unique comparisons, we end up with this result for all of the figures that I showed you previously. So this is a mammoth task within a single paper. All of the figures shown previously are represented here in this biological data set, and this is a model output. And when we just compare the things, well, the little X's show where initially comparisons to controls are incorrect, but you'll notice for each one it's where we've modified two things at once. And in that case, the wild type or the baseline is not really the better comparison. In that case, we really need to look at how trends change relative to, you know, different, we've got auxin along here and we've got sucrose changing along here, so we can see how the relative changes of things happen. When we actually take that one step further, we actually found that the model captured all of the trends observed in the biological data. So that's pretty successful. Now, Nicole comes from an evolutionary biology background, and so she wants to know, hang on, well, what happens if that network's slightly different in different species? What might evolutionary change over time look like? And PSoup lends itself really beautifully to focusing on, instead of playing with parameters, but instead we can play with the network structure. So we have 4 different network structures here, little differences, you know, here and here. And then when we compare the models to the whole data, well, we can see different patterns emerge from these models for just showing 2 relationships, sucrose and auxin, the same ones that Brody used earlier. You can see different patterns that are coming out. You can distinguish differences here and here that differ among the models. And what we actually find using this approach is that actually model 3 and 1 being the birth fleet model, 3 being a slightly different model, which has another arrow here, both of these models actually fit the whole data set, as well as fitting the tested data set that was used by Berthelot. So I think that brings another perspective of the way that this approach can be used. But you're saying to me, I'm sure, this is just really quite a little toy network. It's a bit too small. So, you know, come on, get a bit realistic. So Christos Mitsanis joined the centre as a PhD student, and he was tasked with actually creating a PSoup model for the entire branching network as we understand it. And he actually produced this network, which I'll explain to you how he did that and how good it is. And so basically he started off with a literature review. He narrowed down to 230 papers that were sort of on topic, from which he could produce initial knowledge graphs or networks, encode those then with PSoup and do simulations. And what's really important now for me is to explain to you how we actually test that. So he has to identify some training data. So he chose to use shoot branching and tillering as keywords, among a few other things, and came up with 51 papers that had the following properties. They had within them information that fell into this category: node, perturbation, phenotype. So node, such as I've shown you before, perturbation, which we can do through the modifiers, and phenotype, which is the phenotype of tillering. So examples: gene mutation, branching compared to wild type, shoot tip intact, decapitation, shoot tip, yeah, intact, shoot tip and then decapitation and then branching compared to an intact plant. Wild type plus cytokinin, branch compared to wild type. So that information needs to be in the papers, and there are a lot of papers in, you know, for example, reviews and other papers that didn't get that sort of granularity. And so he ended up with 51 papers that had this kind of information within it. And so the simulations in PSoup are then able to compare for each node data of this type, which he stored in an Excel file and then associated the information from the PDF that he utilised. So he'd go in there and click on it, check he got it right, and every time he was iterating, he could check how he's going. And so basically the idea is if it's not really very good, then you refine the network and you keep going around in this loop until you feel you've reached a plateau where you can't any longer improve the model. And that model now is a network that best fits your training data. And so that's the model he produced here. It has 32 nodes. It has 56 edges, and the edges are the connections or the effects of, say, sucrose with shoot branching, a direct effect, auxin in the bud, direct effect on GA. Those are called edges, and these things with the writing are called nodes. So we've got 32 and 56 and 32 equations, species across diverse species, I think across those 51 data papers. And in terms of the accuracy, so I mean, I guess it's a clunky way to present this, but here are the results sorted now for, you know, sorted against colour on the biological. Again, we have the biological data and the model data, and you can see the different perturbations. This would be a perturbation in that gene, for example. And on the right, you can see decrease. On the left, you can see increase, and yellow is unchanged. And we can see with the X's where there were problems with not with poor accuracy, where the comparison control is incorrect. So the accuracy turns out to be 84% with 79 unique perturbations. And the areas of misalignment are actually areas where I would have said already that there would be problems where I, as a researcher, am not happy with the model. More research needs to get done. So this really is highlighting areas of the network where we really don't understand how it works. So some people in the audience will say, oh, that's great, you've trained your model on the data. How do we know? Can you predict into a different set of data? And I'm not sure if that paradigm is going to be great if we want to actually build networks across all of our knowledge. But in order to test PSoup, I think we do need to do that. So the testing data set we chose was bud release and gene expression. So the bud release is a set of data which is always talking about really early stages of growth, maybe buds only a few millimetres long up to, you know, a few centimetres. So, and we call it bud release. And a lot of gene expression data, for example, BRC1 gene expression, that's TB1 in maize, you know, strigolactone gene expression, so on. And here we have, in this data set, there were 135 unique perturbations, and the accuracy was still pretty high at 71%. And again, the areas of problem were, as we saw before. Plus, there was another problem, which was when you're looking at gene expression, they're often after a treatment. And so a lot of these ones are where we've added a hormone treatment and got problems with gene expression. That could easily be due to timing issues. But we are just presenting here to you the raw results, and accuracy is still very high at 71%. So I think it is useful. It can be used across diverse data sets, even across different laboratories, different systems. But because we're using node perturbation response, it just is a really good leveler to be able to do that. Okay, so in summary, for our model, which we have either 84% or 71% of an untrained data set accuracy. And I can also say, just to reiterate, that this method is scalable. It's deeply evidence-based because we know exactly why each and every relationship is there, and we can access that. It's obviously able to help us think about our current understanding, and it's able to be shared with non-experts in a list of mathematical equations that are directly derived from this diagram with no fudging or fiddling around. So that means it's very transparent for the recipient of those equations. So, okay, we've got a list of these equations, and we've built these mathematical networks. So how can we actually make them useful for a breeding pipeline? So two areas here that I'd like to address. One of them is in relation to using crop growth models. So in the case of a crop growth model, we're talking about a system that captures an incredible amount, potentially a credible amount of knowledge around the plant's growth, different nitrogen, light environments, weather patterns, and so on. And it's usually fixed for particular cultivars, if you like, and it struggles to bring in the genetic component. It's particularly useful for complex traits. For example, tillering. You know, you can have a plant which tillers beautifully under well-watered conditions and has extremely terrible low tillering under drought conditions, or maybe it has, and then this affects the yield dramatically. So highly plastic traits and complex traits. So with that sort of background, you know, Mark Cooper and Graham have been working for a long time. I'm trying to think about how to innovate and make use of all that potential knowledge of the physiology of the plant to improve the breeding pipeline using crop growth models and ultimately connecting whole genome prediction with crop growth models. And one of the goals of the centre is to do that with genetic networks, and in our case using APSIM, which is a crop growth model established by Graham and colleagues over the years, used a lot with sorghum, which is an important crop for coffee. So we often talk about the branching network as an example that could be utilised within this pipeline. And so our goal is to try and connect that branching network with APSIM. So if we just zone in on a tiny part of APSIM which has the tillering aspect, it looks something like this. We have environment, temperature, photoperiod, radiation affecting the sugar supply, and also it feeds into the supply-demand ratio. So you've got supply on one hand, but you've also got your sink strength and demand on the other. And that's calculated within APSIM, and that actually will predict the potential tiller number depending on variety. Also, you've got management, row density, and the like. So our goal is then to connect our modelling here. Of course, the supply-demand ratio is a little bit like thinking about the sugars. So what we did was, what Christos did with Graham's team, was to connect the supply-demand ratio through to sucrose in the PSoup model, or rather connect the PSoup model with APSIM using the output of PSoup, or the input from APSIM into sucrose. Sucrose goes into PSoup, and then PSoup can output the branching. And the modifiers can be used to regulate the genotypes. So now we have a genotype environment management paradigm happening with a connection of PSoup and APSIM. And I sort of alluded to this before, but if you've got different supply-demand ratios because you've grown plants under different environments, and you can have then different varieties which will have different, they're still showing an upward trend, but they start at different positions. And so this is the observed data in the symbols, and then the APSIM model with the lines. And if we instead of using APSIM's best fit to the data, we can think about using PSoup to produce those trajectories for different varieties. So these are different genotypes that have been introduced into PSoup with the modifiers, and then you can see how PSoup would cause different responses according to the supply-demand index. And so this is a good theoretical framework to think about how to connect the genetics into APSIM so that we can have a G by E by M model. the other area the Centre's been working on, another one that I just want to highlight, was improving genomic prediction, which Shinichiro has been doing, Mark Cooper and others, Mel Wilkinson, producing an ensemble method. And I'd just like to point out that within that ensemble method, there's the graph attention network, which is a network method, and that we're hoping in the future that we can connect to that with the mathematical descriptions of the network, so that we can use our networks within that context. And we could also think about doing that with GWAS as well. The other thing we'd like to use the networks for, I'd particularly like to do this, is to help them interrogate large multi-omic data sets, particularly RNA-seq, but other data sets also, so that we actually make use of prior knowledge when we analyse RNA-seq data, which is very poorly done presently. So the last thing I've got to cover, I have to be very quick, I think, is designing an automatic network generator using AI. So this is the brainchild of Christos and David, and they've designed a workflow called FlashP, and it's absolutely amazing in my view. It's inspired by PSoup using an agentic AI approach. Because it's a multi-agent system, you can sort of drive it in the way that the expert would drive it. So doing all the things that we've talked about, screening existing knowledge, capturing specific information around node perturbation phenotype, and then checking that it hasn't made anything up, hasn't hallucinated in any way, by actually checking that these do correspond back to the information in the PDFs, and then linking those PDFs explicitly to this information, making it very transparent. Rejecting inconsistencies, as we had done previously. If different experiments give opposing results, they get rejected. And then, of course, you can use a range of different methods to best fit the model to the data, such as algebraic equations or normalised Hill functionalities and RWRs. And just to give you a sense of what that looks like, it's 30 minutes to produce one of these networks. It's 6 months if you do it manually, let's say. This particular one has, it has, around 53 nodes and 84 edges, and its accuracy is actually fantastic at greater than 90% across 165 aggregated perturbations. Compare that back to what I said to you before. We have fewer nodes and edges from the manual analysis, and the accuracy was actually a little bit less. So, that alone is convincing me that this method, which is transparent, is very strong. And I just want to draw your attention to transparency. So this is the software that they've created. This is for the flowering network in Arabidopsis. And if we just click on one of the edges, we can see where the paper is and what the information is that was telling us that full activates leafy in the flowering pathway. So if you want to know more about this, and I surely hope that you will, please contact Christos or David, or if you want me to give you their emails, more than happy to pass them on. Fantastic workers. And they urge me to tell you FlashP is so much more. I've got a lot of stuff in the pipeline. Some of it's finished, some of it's ongoing. But, really this is way more than just a way of pulling out networks. It's something that you can use to drive your own research. You can conduct virtual experiments. You can connect all sorts of layers of knowledge. You can connect it to APSIM, etc. Okay, so in summary, I talked about PSoup and creating mathematical networks. I talked about connecting PSoup with APSIM, and I've introduced you to FlashP as an automatic network generator and much more. And I hope that, folks in coffee will be enthusiastic to imagine how we might be able to use these things in the context of coffee, and I'm really looking forward to doing that both with my colleagues in the centre and in coffee over the coming months and years. And just thank again the wet lab, if you like, few dry lab folks, and I'd like to thank the Centre of Excellence, which for which most of this, well, all of the funding, I think, came for this research. So thank you, Mark.
Thank you very much, Christine. And we have approximately 8 minutes for a series of question and answer opportunities. So as indicated at the beginning, the housekeeping stage, what we wanted you to do was put your questions in the Q&A section, and I'll read out the questions from there. So I'll take the first question that's been entered by Craig Hartner and read this out. What are your ideas about opportunities to translate these concepts to hort tree crops, which are more complex and have less data? Christine.
Yeah. Well, I think this paradigm of using directional change is really useful because you can apply it to crops, you know, outside of your current focus. But you can still bring, say, a small data set that you've got in one species. If that's not incongruent with the network, then we can imagine that the network might still apply to that species. We can also make modifications to the network so that it does apply to that species. So I think, Craig, that we can think about it for breeding and genomic prediction in small population sizes. I think this is a really great way to leverage knowledge that comes from well beyond the species. Yeah, I think that's probably the main focus. Okay. Happy to chat more. I've got sort of other ideas around, you know, architecture and the like of tree crops as well, but yeah, go ahead, Mark.
Thanks, Christine. Maybe I'll take the opportunity to ask a question, since you indicated you had an interest in connecting into the work that plant breeders do. So as a plant breeder, I'm responsible for running a breeding program. And one of the things that I use to design and operate that breeding program is the breeder's equation. And within that breeder's equation, I think there's a clear connection to the type of work you're doing on looking at gene networks, to the sources of genetic variation that we're trying to manipulate over cycles of breeding. One of the questions I think is really interesting to think about is, how do you think the accuracy you're talking about might relate to the heritability concepts that breeders are fundamentally interested in? Because ultimately that determines how you translate your prior genetic knowledge and knowledge of genetic variation into opportunities to direct selection trajectories. So really the heritability of your network, have you given some thought to that?
Heritability of the whole network, you mean?
Yes, ultimately.
I mean, the heritability of, you can certainly show why under field conditions you might really struggle to get a high heritability when you've got all that variation going on, attacking the network from all kinds of different angles. So I think if you can figure out how to connect that environmental variation into your network, then you will certainly be able to reduce the variation, if you like, or count for that variation, which should increase your heritability. If I'm answering your question the way you were expecting there, Mark?
Yeah, maybe just quickly following up on that. In your search of the literature, did you come across any publications where people had asked questions about the heritability of networks that were controlling the traits that are manipulated in plant breeding programs?
No, I haven't. You can send me them, Mark.
Yeah, it was interesting. There are publications out there, so I was interested to see if you'd actually picked those up. in your literature searches?
Yeah, no, I mean, that's why I've got sort of Sean there. I think that this is a capacity there for you guys to utilise, for you guys to think about these questions. I think a plant physiologist thinking about them is probably not the most sensible person to be doing it. I think it needs to be done by experts much closer to the coalface that are working in predictive technologies a bit more than I am. But, you know, I like to think that I'm helping to build the teams with you and others to do that. And I think what we need to be doing is getting those individuals to be giving these talks.
Okay. Yeah, no, thanks very much. I'm sort of not seeing any other questions in the panel, and I guess maybe for the final question I'll ask it, and I'll return to the question that your parents asked you as you discussed at the beginning, and that is, for you, what would you see as impact from the work that you're actually doing? How would you judge impact?
Yeah. Look, I think this way of thinking about you know, the model from a developmental perspective with a baseline control and relative change to that control, and being able to scale that across all prior knowledge, not just you know, data sets that are highly relatable. I think that that's a huge contribution to thinking about how to scale up these networks. And I think success in being able to pass that through would be around whether those can actually be taken up. Whether, for example, Sean can use that knowledge in a graph retention network, which is embedded into an ensemble framework, for example. If he can achieve that, and I can say, right, well, we've managed to find a few more SNPs or enhance their heritability or that sort of thing, then I'll be like, I've won.
Okay.
I'm actually doing something useful. That would be one example.
Okay. One final question from Peter Waterhouse. And so I'll ask his question. With the networks, have you got a report from the lists of the components of your network, Peter's referring to, of the most significant genes that would be the most beneficial targets to, say, increase another quantitative trait such as yield in a given location and environment? Yeah,
so I think that's the vision of Flash P, is to, because it's agenic and because it's you can sit in there and you can sort of in a range of frameworks from very guided to very unguided. You can ask those sort of questions. For example, you could say, if I want to CRISPR a sorghum for a particular environment, what would be my best target genes to I don't know, regulate tillering to enhance yield under a particular environment? It would be able to give you, I would say, a world's best guess at what those genes would be, and with evidence, not so to be with reasoning. Okay, and not only that, but connecting the tillering network with all those components that sit underneath APSIM, all those trait components, you can actually do that in the context of the whole system. And I'm not saying we're there yet, but I think we're very close to being there. I think current challenge is going to be around validating these predictions. But yeah, the fact that the predictions are already tested against all current knowledge is a heartening aspect of it.
Okay, thanks very much. And I think that'll be our last question. Probably we'll finish up with an answer that gives us the world's best guess, which is probably a good place to ask you. Look, everybody, this is a very well-attended seminar, and thanks to everybody for joining us today and listening to Christine's presentation. And on the screen, you'll see a QR code where, if you haven't already, please sign up to the seminar invitation list so you can and be notified of future seminars. So with that, I will bring the coffee seminar of today to a close and thank everybody for joining us, and particularly thank Christine for a very thought-provoking and forward-looking synopsis of a very significant amount of work by Christine and her team. So congratulations to Christine, and I'll just say thank you to everybody, and we'll close the seminar there. So goodbye.
Thank you, Mark. Thank you. Bye.